Welcome to the third chapter of our macromolecule series, Nucleic Acids. Nucleic acids, such as DNA and RNA, are the instructions for building proteins. This is super important for life because most of the building structures of any body are protein, and enzymes help conduct the chemical reactions in our cells that keep us alive. And when I mean they conduct the chemical reactions, I mean just about everything. The reason why we can see is because there's a very specific protein that interacts with light and then changes its shape, interacts with another protein, which ends up sending messages to the brain, like a very long chain of chemical reactions. And most of those chemical reactions are altering proteins or conducted by proteins. So nucleic acids are the instructions for all of that. We need them. We've got two types of nucleic acids. We have DNA and we have RNA. DNA is the classic double helix molecule that spends its time in the nucleus. And it does that because it's the original information. It's the source material. We've got to keep it protected in that nucleus because out here in the cytoplasm, there are tons of chemical reactions going on. And if that DNA were to get damaged, we wouldn't be able to make proteins. The cell wouldn't be able to do its job and things would be bad. So we got to keep that DNA protected. So that's what we do. We keep it in the nucleus. Now, but we actually need to read the information, right? The DNA's got the information, we need to read it. So we make a copy of that information, and that is the RNA. But because it's just a copy, we can make it again. Who cares? Send it out of that nucleus, get it out there to a ribosome who can read that information and start making protein. So that's the story in a nutshell. DNA is the original information for proteins, which our body runs on and is built of. RNA is a copy of that information which can leave the nucleus because it doesn't need to be protected like DNA does. You go to a ribosome, you start making protein. Great. Now, let's talk about nucleic acids in terms of monomers and polymers. And we have to move me, so okay. The polymer, RNA and DNA, DNA is really long. DNA is one chromosome is made up of one long strand of DNA, and it is 250 million nucleotides long. The longest chromosome in human cells is 250 million nucleotides long. The shortest chromosome in human cells is about 50 million nucleotides long. So we've got quite a range, but that's a lot of monomers. So the monomer is a nucleotide, and the polymer is either DNA or RNA. And so we link these nucleotides together in those, 50 million chains to make our nucleic acid. Each nucleotide comes with three parts: a phosphate, a sugar, and a nitrogenous base. Now, every single phosphate in every nucleotide, be it DNA or RNA, is exactly the same. It's a phosphorus atom bullied by 4 oxygen into sharing more electrons than it normally would because of the difference in their electronegativity. So this nucleotide right here is linked to this nucleotide right here by that phosphate. It sort of forms this backbone. They call this phosphate-sugar link the backbone of your DNA. So that brings us to the sugar. The sugar is a 5-carbon sugar, and unlike the phosphates, there are varieties. There are 2 different sugars that are included in nucleotides. One is called ribose, and the other is called deoxyribose. If we start over here with ribose, we can see, yep, it's a 5-carbon sugar, and it's got this hydroxide group hanging down on this carbon here. That is our ribose, and this is the sugar that is always present in every single ribose nucleotide. Well, if we take the oxygen away, if you deoxify that ribose, you would get deoxyribose. And this is the sugar that is in every single DNA nucleotide. So if you've got DNA, you've got the phosphate, and you have a deoxyribose. If you have RNA, you still have the same phosphate, but then you have a ribose. And that's where they get their name. The D of DNA is deoxyribonucleic acid versus the R of RNA, which is ribonucleic acid. Now, the nitrogenous base are the interesting parts. They're the part where the information is. And so they are different. There are 5 bases to choose from. It turns out that uracil is only present in ribose, and thymine is only present in deoxyribose. But we'll deal with that later. You don't have to know that now. What you need to know about the nitrogenous bases are that they come in two flavors, the purines and the pyrimidines. The purines are double-ringed molecules made of carbon, nitrogen, and a little bit of oxygen. And the pyrimidines are single-ringed molecules made of carbon, nitrogen, and a little bit of oxygen. There's much more, so we'll have to do a nucleic acids part 2. Hope this helps. Struggle well.